Alrighty guys, welcome to 10.3. This is our last video and we're going to use this to take a closer look at the mitotic spindle and the other factors that are going on during mitotic division. So a big part of this is going to be us taking a look at the components of the cytoskeleton. So when you finished video 2, I told you to go ahead and try some of these next questions that we are going to talk about involving the cytoskeleton. If you haven't done that, pause, go ahead, give them a try, come back, and now we're going to talk about them. All right, so the mitotic spindle is going to be those microtubules that we keep talking over and over about. They're really important because these are what the centrosomes do, and the organization of the microtubules all at a central centrosome is actually really critical. It's what allows us to have these poles at the two ends. So microtubules will polymerize and depolymerize, meaning they will form and deform, but they all originate at that centrosome. So all of the fibers that are branching out have these poles that organize them. In prometaphase, some of those spindle microtubules will attach to kinetochores of chromosomes, and others are going to actually attach to the fibers of opposite centrosome. So the idea being some of them are responsible for the tugging back and forth that aligns all of those chromosomes in metaphase. And remember, if you're not attached to both poles, you won't actually reach the middle. So it's an important checkpoint in mitosis. But others are going to overlap so that you can push that spindle apart. And this is necessary once you've separated those sister chromatids by dissolving those cohesins, cleaving those cohesins that hold them together. So in metaphase, again, that push and pull is responsible for lining the chromosomes up. These are kinetochore microtubules. And then you're going to see these in anaphase, this idea of non-kinetochore spindles that actually elongate this entire network.
Watch how microtubules assemble to form the spindle fibers during prophase of mitosis. These time-lapse images of cells expressing a green fluorescent protein tubulin fusion were taken using confocal microscopy. Notice how the centrosomes at the opposite ends of the cell act as nucleation sites for the growing microtubules. Credit: Ronald Bale, University of California, San Francisco.
So here's a cartoon. Again, the metaphase plate is an imaginary plate. There's not actually a plate of glass inside the cell. But hopefully you can see here very nicely the idea that this metaphase plate only forms and that we only have this alignment because of the nature and the dynamics of these microtubules. As they form, they push. And as they deform, they pull. So this push and pull, push and pull. This is really about that process of polymerization. And this is achieved through dehydration synthesis. All right, so take a look down in the bottom right. This is a picture of kinetochores. Kinetochores, proteins, motor proteins that actually attach the centromere to this microtubule. So remember, motor proteins are capable of walking. They can move things. For example, we have dynin, which is capable of moving along microtubules, of walking these vesicles. Well, kinetochores are capable of moving along microtubules, but instead of pulling a transport vesicle along, they're going to pull a sister chromatid. So these motor proteins can walk along these microtubules and carry with them sister chromatids. This happens during anaphase, and there's an important thing where as these sister chromatids walk backwards, they can depolymerize microtubules. So remember that when we push and pull, we go through this process of creating and deforming. There's not actually muscles on either side pushing and pulling. It's simply the process of when you go through and create it, it gets longer. And when you depolymerize it, it gets shorter, and creating and depolymerizing and creating and depolymerizing. But once you've lined up in the middle in order to separate them, you go through the process of depolymerizing on both sides. Kinetochores not only walk these backwards, but they actually depolymerize tubulin behind them. Now we're going to take a closer look at cytokinesis. So cytokinesis in animal cells is going to be the result of actin. Now this is different for, let's say, a plant cell, a walled cell. We're really talking about non-walled cells here. But actin filaments, because they form that cortex along the outside, they're perfectly poised to be able to begin to constrict in a particular area. When they do, we form what's known as a cleavage furrow. And I will allow it up to each of you how you would like to remember exactly how this is called a cleavage furrow. But I figure you can probably work something out. Those microfilaments are contracting around the cytoplasm like a string, pulling, 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 and eventually, boop, pinching it into two individual daughter cells. Telophase, which is that reformation of the nuclear envelope, and cytokinesis actually happen pretty simultaneously. So oftentimes you're going to see this name telophase and cytokinesis together. It's not because the two require the same process or network, but it's really about the fact that timing-wise in the cell, they happen kind of at the same time. So, if you haven't already done so, please answer these questions. In which way do microtubules contribute to mitotic division? Hopefully you told me that these are the mitotic spindles. And microfilaments? Yeah, these are cytokinesis. Lastly, intermediate filaments? This would be the nuclear breakdown and reformation. All right. So let's get into this idea of cell cycle checkpoints. I've actually used this word before. Remember how we talked about in G1, if you didn't have a signal to go forward and divide, you wouldn't be able to pass that check. And so you'd kind of go into a resting phase. It would be the I'm not ready to have a baby yet phase. I just want to go to college and figure out my life phase. You really need a signal, right? You need a signal that says, gee, I want to have a kid. It's not something you do on your own. Well, that's kind of like a checkpoint. These cells need to have a signal to go forward. So we're going to talk about three different checkpoints. The first one is the G1 checkpoint, and that's that question of, do I want to have a baby or not? Do I want to kind of stay stationary, just stay the way that I am? And that's actually an extremely important checkpoint because it kind of helps to prevent cells from just going off and dividing willy-nilly. There's also a G2 checkpoint, and although we're not going to talk as much about the G2 checkpoint, it's still really important. Prior to the cell actually going through the process of division, it's like an actual checkpoint. The cell is going to survey and make sure that everything looks okay, that we didn't accidentally make way too many of something, or that we're not trying to go through this without actually duplicating our DNA, which would be a really bad thing, and so forth. The M checkpoint during mitosis is actually the metaphase plate. So even though we don't refer to metaphase as a checkpoint in and of itself, it really is. So you can't go through the process of cleaving those cohesins in anaphase unless you've actually had that alignment of all the chromosomes on the metaphase plate. This checkpoint ensures that every chromosome is correctly attached to both spindles, so that when the cohesins divide, the chromatids split. If your chromosome is only attached to one spindle, well then when the cohesins divide, it won't change anything and you will simply take both chromatids into a single daughter cell. So let's talk about one of these checkpoints. So here's, for example, this M or mitosis checkpoint, which happens in metaphase. A cell in mitosis will receive a stop signal when any of its chromosomes are not attached to spindle fibers. Remember, signals are actual chemical signals. So we're going to talk about these in a minute. But there are proteins that are actually made. There are chemical signals that are actually made and says, something's not right, stop, don't go forward. You have to have all chromosomes attached to spindle fibers with both poles and a go-ahead signal that allows the cell to proceed by digesting the cohesins and allowing these sister chromatids to separate. I want to talk about a particular component of cell cycle, cyclins and cyclin-dependent kinases. These are extremely important in this traditional G1, G2, and M checkpoints. Cyclin concentrations change based on cell cycle process. So as you go through, as you get progressively farther through the cell cycle, you're going to see the accumulation of cyclin. And this in this graph is shown in purple. What you'll notice is that cyclins in this particular one, this example, so this particular cyclin, is going to accumulate after the replication of DNA in the S phase. So these are accumulating during G2 phase and into the start of M phase. But during this mitotic phase or M phase, cyclin-dependent kinases, what do kinases do? Oh yeah, they move phosphates. These cyclin-dependent kinases, their activity is going to rise. And this activity is going to be dependent on the nature of this excessive concentration of cyclin. So you can imagine that now this kinase has an activity, but that activity is only going to occur during a particular cycle because we had to accumulate sufficient amounts of cyclin. So what do I mean by this? Well, take a look at this entire cycle. I want you to start by looking at the cyclins themselves. That's the thing in purple. And if we start at G1, or light green on the left side, you'll notice that there is no cyclin. The cyclin was degraded following the cell division. So our newly divided cell has very little cyclin. And the cell doesn't receive the signal to produce more cyclin until the DNA has been duplicated in the S phase. And it's going to begin to accumulate this cyclin. As that accumulation begins to occur, you're going to see an association between the cyclin and the cyclin-dependent kinase. Listen to the name. It's a kinase dependent on cyclin, okay? When that cyclin-CDK complex comes together, now we have a kinase with actual activity. So the cyclin-CDK complex can phosphorylate signaling molecules that lead to cellular responses. For example, you can lead to the degradation of cohesins, which leads to an anaphase. That's a signal and a response. When the cell actually divides and mitosis is complete, the cyclin becomes degraded, the signal goes away, it's a reversible signal, and we go back to G1, where there's no cyclin accumulation, which means that there's no cyclin CDK complexes, which means we're not doing the job, not sending the signal, and so you're not going through mitotic events. So there are two types of signals, go signals and stop signals. The presence of a CDK cyclin complex, a kinase which is doing things, is an example of a go signal. But this isn't the only kind of go signal. For example, this G1 checkpoint. If your cell is not ready to divide, it's not ready to have a little cell baby, then it's going to exit this cell cycle. It's going to go off, and the lack of a signal, right, serves as a stop. If there's not a sign from above that it's time to have a child, then it's probably not time to do so. But there are going to be signals known as growth factors. Growth factors like epidermal growth factor and like the hormones that you got as a teenager, they tell cells to divide. So your bone cells, as you're growing, were receiving lots of growth factor, and that growth factor was important. It took cells out of G0 and sent them into the synthesis phase and said, go duplicate. I need more bone cells because I'm a growing boy. Now, healthy cells only receive this signal when you have the actual production of chemical signals that are growth factors. Healthy cells are going to stop when they become crowded. Unless you're actually growing in size as an organism, there's not going to be enough space for them. They're going to stop. It's called density-dependent inhibition. So as it becomes very dense, there's an inhibition. Cancer cells respond too well to stimuli, like growth factors, and they ignore inhibitors, like density-dependent inhibition. So Growth factors are examples of go signals, and cancer just says, all I want to hear is the go signal. And density-dependent inhibition is the stop signal. And cancer says, I can't hear you, I can't hear you, and it divides anyways.
Our bodies are made up of 100 million cells. They're so small, you can fit 100 cells on the top of a pinhead. Most cells have a nucleus, cytoplasm, and cell membrane. The nucleus is the cell's control center and holds the cell's DNA. Your DNA carries all the instructions needed to build your body and maintain its functions. Each instruction is carried on a unique piece of DNA called a gene. Cell growth and multiplication is part of a continual process called the cell cycle. To move through the different stages of the cell cycle, each cell has to go through a series of checkpoints. These checkpoints act a bit like traffic lights, and they only give the green light to a cell that's ready to go through to the next stage in the cycle. Only healthy cells are allowed to multiply, and so the cell cycle must control which cells are allowed through. The DNA inside cells can sometimes be damaged, either by mistakes in the normal processes in our cells or by things in our lifestyles or environment. Damaged cells are stopped at checkpoints and either repaired or destroyed. But occasionally damaged cells can find a way to sneak through. And sometimes the faulty DNA instructions may tell the cell to multiply at the wrong time or in the wrong place. If a cell accumulates too much damage over time, it can become a cancer cell. They then multiply out of control to form a lump or tumor. This is how cancer starts.
All right, guys, if you guys are really interested in cancer, and I know many of you are, then I want to make you aware of a couple of things. The Emperor of All Maladies is quite a large book, but they actually created a documentary from PBS, and it's free if you have Amazon Prime as well. You can watch the intro for free no matter what, but it's a fantastic documentary on the history of cancer. Not only is it going to tell you a little bit about what cancer is and cancer treatments, but it's going to talk about the societal impacts of cancer. Things that you probably don't remember because from the time that you were born, everybody was out doing, cancer walks and raising money. But it didn't always used to be that way. The Immortal Life of Henrietta Lacks is case in point. This tackles the morality of cancer rights and research. Henrietta Lacks was a woman who had ovarian cancer, and she unfortunately perished of her disease. But prior to her death, we took cells from her cancer, and those cells are still growing and dividing in labs all over the world. I personally have used cells that were descendant from Henrietta Lacks' cancer many times in my own research, and yet her family has never seen a penny from all of the good that it's done. Her cancer cells have been sent off into space. They were used to develop the polio vaccine, and there are the images that we show you 9 times out of 10. They're everywhere. There's a book by Rebecca Skloot that is fantastic and wonderful. But if you guys are more video people, there is an HBO film that's starring Oprah Winfrey as Henrietta's daughter, Deborah, who recently passed away. And it's a fantastic opportunity to think about this not only from a researcher standpoint, but also from a moral standpoint. All right, guys, I want you to go back and work through this. As you know, that SymBio is extremely helpful for helping you to kind of work through these cycles. So I strongly recommend that you do the SymBio activity before I see you on Thursday. But the due dates for your SymBio haven't actually changed. So I want everybody to work on it, but know that you aren't magically going to have to turn in assignments prior to when you would have. It's just that we've made it available to you. I'm going to see you in recitation, so be prepared. And have a notebook because you're going to want to draw these cells as they go through the divisions. So on your recitation, I'm going to be asking you guys to submit some drawings as well. So you'll want to think about how, as you work with your groups in recitation, that you can actually do some drawings. You might find that the whiteboard function on Blackboard helps you to do this, or you could have someone print out the recitation to do the drawings, and then you can submit those pictures. Just be sure that you always submit a single document for recitation. So maybe what you can do is take a picture of and then actually insert that image into a doc file. If you guys have questions or concerns, you know where we are and you know where you can find us. We'll be available in office hours this week and going forward. As always, please let me know what things are working for you guys and let us know what's not so that we can help you where we can. All right, see you in class.